Hello everyone, I'm Haili. It's my great honor to attend this meeting. I'm from the Pharmacy Department of Chinese PLA General Hospital. My teacher is Professor Guo Daihong. Today, I would like to share with you my research. Analysis of clinical characteristics and automatic monitoring of drug-induced arrhythmia in 167 546 inpatients. During the global pandemic of COVID-19, some people questioned its drugs, such as lopinavir, ritonavir, suspecting that these drugs may increase the risk of arrhythmia. Because studies have shown that one of the most frequent complications in medicine is sudden death caused by drug-induced arrhythmia. Drug-induced arrhythmia, especially QT prolongation, is one of the key adverse events to monitor in new drug development and post-marketing drug management. The ICH, SNV, and E14 guidelines require cardiac safety assessment before new drugs are marketed. However, due to the limited population of clinical trials and the short trial time, there are still some potential risks of cardiac adverse reactions that haven't been discovered, leading to the withdrawal of some drugs from the market due to serious arrhythmias, such as esomeprazole, sulfadoxine. According to a scientific statement of drug-induced arrhythmias issued by the American Heart Association in 2020, a variety of commonly used drugs may cause diverse sorts of arrhythmias, including QT interval prolongation. To 30 points. A review of the literature shows that drug-induced QT prolongation is a well-known adverse effect, but the public has limited knowledge about other types of drug-induced arrhythmia, such as heart block. There are few and incomplete studies on drug-induced arrhythmia in China, and there is also a lack of large sample real-world studies on the incidence of arrhythmia caused by common drugs. Therefore, we conduct these studies. The purpose of this study is to use the text classification technology of ADE-ASS2 to mine the unstructured clinical narrative of drug-induced arrhythmia in our hospital and analyze the demographic information, clinical manifestations, drug distribution, and incidence related to affected hospitalized patients in the real world, thereby providing a reference for safety in clinical drug use. ADE-ASS is an automatic monitoring software for adverse drug events developed by Guo Daihong's team in 2012. Today, automatic monitoring studies of millions of inpatients have been completed, including liver injury, acute kidney injury, allergic reactions, which provides support for clinical medication and pharmaceutical management. Compared with the traditional spontaneous reporting, the automatic monitoring of adverse drug reactions is more efficient for post-marketing monitoring of drugs, and the strengthening of pharmacovigilance mirrors. The main operating process of the ADE-ASS2 was as follows. First, a monitoring plan was established, including the monitoring time, age range, and the module. The arrhythmia module use text classification technology to identify keywords. The keywords of arrhythmia were searched in electronic medical records and electrocardiogram. After repeated testing and verification, the module runs stable. Second, the inpatients were monitored by the module. Once the monitoring indicators triggered the keywords, the system could identify it as a suspect case and issue early warning signals. The arrhythmia module of the ADE-ASS2 can be used in ADR studies in the real world, and about 81% of irrelevant cases were automatically eliminated in this study, which improved the efficiency of the research. Third, the suspected cases were manually evaluated by two clinical pharmacists based on the inclusion and exclusion criteria. The positive cases were determined. The clinical pharmacists have undergone uniform professional and technical training. First, we collected the information of positive cases from electronic medical records, including demographic information, diagnosis, suspected drugs, ECG and laboratory results. The ECG results of our hospital were reviewed by physicians in the ECG department. Fifth, we made a statistical analysis of the data. The results are as follows. Result 1 is the basic information of positive patients. There were 2,097 positive cases, including 1,234 were male, 863 were female. The incidence was 1.31% and 1.17% respectively. In this study, the difference was significant. The median age of positive patients in this study was 64 years. About 47% was over 65 years old. By intergroup comparison, the age was associated with the occurrence of drug-induced arrhythmias. In addition, the findings of this study showed that emaciation, obesity, and underlying disorders were associated with the occurrence of drug-induced arrhythmia, which stressed that clinicians and pharmacists should assess patient-related risk factors when using drugs that are prone to induce arrhythmia. Result 2 is the distribution of ADR, incubation period, and hospitalization time of positive patients. The median time was 4 and 14 days, respectively. There were 45% of ADR occurring within 3 days after admission, and 94% within 15 days of admission. This data suggested that the majority of patients with drug-induced arrhythmia could be closely monitored while hospitalized. Result 3 is the trip of arrhythmia in positive patients. Of the 2,097 positive cases, 288 had QT prolongation, 1,809 had arrhythmia. 49 of the arrhythmia were combined with QT interval prolongation. The incidence of arrhythmia and QT prolongation were 1.08% and 0.2% respectively. In the process of risk factors, QT prolongation is easily transformed into TDP, which then developed into ventricular fibrillation, leading to sudden cardiac death. A QTc interval over 510 milliseconds is strongly related to TDP and cardiac arrest. There were 672 cases of arrhythmia with pre-medication ECG alterations, including 109 cases with QT prolongation. This study found that pre-medication prolonged the QT interval, which may increase the risk of drug-induced sinus tachycardia and atrial fibrillation. In addition, ECG alterations before medication were associated with drug-induced extrasystoles and sinus bradycardia. Result 4 shows the relevant drugs and their incidence. The 1,809 drug-induced arrhythmia involved 176 kinds of drugs in 13 categories. Most drugs for the cardiovascular system, anti-infective drugs. This is basically consistent with the results of other related studies. Cardiovascular drugs, including many categories, and the incidence of the drug-induced arrhythmia varies from 0.77 to 9.49%. Most anti-infective drugs are fluoroquinolones. Fluoroquinolones use was linked to an increasing risk of several arrhythmias. There are researchers show that it increased the risk of arrhythmia by 85% and the risk of mortality from cardiovascular disorders by 71%. Of the fluoroquinolones, the most cases is levofloxacin in this study. The incidence of levofloxacin-induced arrhythmias and QT prolongation were 1.24% and 0.44%, respectively. However, there are limits of our study. First, the module is a single-center study without external verification. Due to difference in patient characteristics and clinical narrative in different health institutions, that may reduce the accuracy of the model. Second, the degree of the arrhythmia was not detailed. We didn't analyze the increase or decrease of heart rate after medication. Third, this study is retrospective. The results need to be verified by a case control or prospective study. The final part is the conclusion. The real-world incidence of drug-induced arrhythmia in this large sample of inpatients was in the range of common, and the QT interval prolongation was an occurrence occurrence. Patients with risk factors should avoid drugs that cause arrhythmia. If necessary, electrocardiograph should be monitored for at least 3 days. That's all what I'm sharing today. The article has been published in the European Journal of Clinical Pharmacology. If you are interested in, you can read it through the following link. If you have any questions, please email me. Thank you.